by determining structures more easily and getting better models. This presentation is available online at the website that I just highlighted, phoenixonline.org presentations, in case you want to follow along and didn't download it already. It'll be there afterwards as well. So let's talk about this. AlphaFold models. I'm going to give you the 4 bullet point summary of this talk now. AlphaFold models are great hypotheses for protein structures. And if you don't learn anything else from my talk, just remember that. They're great hypotheses. They're not answers, they're hypotheses, and they are great. The second part of my talk is going to be, you can jumpstart structure determination by X-ray, Cryo-EM, and other techniques using AlphaFold models. They're really great for this. Third, and you probably don't know this yet, you can improve prediction by AlphaFold if you have some experimental information. And I'll show you how you can do that. And all of this allows a new workflow for structure determination by Cryo-EM and X-ray. And I'll give you an example of how you can do that. So let's go. AlphaFold models are great hypotheses for protein structures. Let's see what we mean by that. So let's talk for a moment about where AlphaFold models come from. I'm not going to go into detail about how AlphaFold works, but we are going to talk about what information goes into AlphaFold, generally how it works, and therefore what can you expect it to produce. So AlphaFold is one of a number now of machine learning prediction tools applied to protein structure. Basic input, a sequence of a protein. Constructed from that, typically automatically, is a multiple sequence alignment showing the alignment of related proteins and all the differences or similarities of amino acids. Along with this goes, in training, a known 3-dimensional structure taken from the protein data bank. AlphaFold is trained on some 180,000 protein structures in the protein data bank. And the training leads to some 21 million parameters, an astounding number of parameters. When we move to prediction, we have the same starting input, a sequence for your protein, multiple sequence alignment for your protein, 21 million parameters, and then a process which involves focusing attention on important relationships in the protein, which we'll come back to later, that yields a 3-dimensional prediction. Notice that the prediction is not exactly the same as the structure. This particular structure we're going to see throughout this presentation, and you'll see how all these things work generally with respect to this particular structure. Along with the 3-dimensional prediction comes a confidence estimate for every residue and for the protein as a whole. It's referred to as a p-predicted LDDT that basically refers to how accurate that protein structure is, and the P is the prediction of accuracy. This is a novel thing in the protein structure determination world. it's going to be very helpful in what we're going to work on today, what we'll see today. Okay, now one of the key elements of this information going in is that multiple sequence alignment. The multiple sequence alignment tells you, or tells AlphaFold, two really important things. One is what residues co-vary when you compare sequences of related proteins. So the red ones here tend to change together in different copies, in different sequences that are evolutionarily related. That often means that those residues are close in three-dimensional structure. That is, residues that are close, touching, might tend to change together because one might compensate for the change in the other. Doesn't always happen that way, but they tend to co-vary. So covariance is an indication of distance between residues, and that's an enormously powerful piece of information about structure. A second really important aspect about multiple sequence alignments is that if you have correctly aligned the sequences of a set of proteins, all of which have similar structure, all the sequences in the alignment should be compatible with the right structure. Now, AlphaFold is not programmed to interpret any of these things that I just showed you. AlphaFold learned how to analyze sequence alignments. We don't actually even know if it does what I just told you, but it seems reasonable to suspect that it does, since this is the information that is present. Now, since residue covariation is very important piece of information, as is the fact that all the sequences must be compatible with a single structure, if you have more sequences in your alignment, then you tend to have more information about the structure. So here, this plot, this is for the same little structure I just showed you. The left-hand panel shows on the y-axis is the number of sequences that are aligned, and the x-axis is residue position. On the left-hand side, there's tend to be many more sequences in the sequence alignment. On the right side, there aren't so many. And then if we move over to the right-hand panel, the predicted confidence by residue position on the left is very high. Notice it's in the 90 to 100 range. And then as we move off to the right, where the sequence number of sequences goes down, the confidence drops. Then actually it comes back up. So the confidence is not directly correlated with sequence coverage, but it's related. Now, how does this confidence turn into a comparison of accuracies? So here are the two structures I showed you actually. 7MJS is the PDB identifier of this particular structure. It's in pink. The AlphaFold prediction is in green. Notice that part of this is quite similar and part is not. The left-hand side is the part that I showed you that has a very high sequence coverage. It has very high confidence. That's residues 1 to 100 here. And as you can see, the structure is very similar in the prediction and in the actual structure. The right-hand side has relatively lower sequence coverage, although not tremendously lower. It has lower confidence and it has lower accuracy. Accuracy of the well predicted or high confidence part can be quite good. So this is looking in the core of that left-hand side of that structure. Many of the residues superimpose within tenths of angstroms. Not everything is identical, but it's an amazingly good prediction. Oops. So high confidence residues can be very accurate. Now, what about limitations? So we saw what information goes in. What can we expect about what's going to come out? Well, first, it's just protein. And this is a big limitation. There's no water, there are no ions, no covalent modifications, there are no carbohydrates, no ligands, no DNA, no RNA. So all those things you do not get to predict today with AlphaFold or with any of the other techniques, but you will down the road, but not today. So that's a big limitation. Another one, it's trained on good and poor structures. So today, AlphaFold was trained on 99% of the PDB, of the proteins in the PDB, only excluding very, very like greater than 9 angstrom structures. So that means that a structure that's not so good that's in the PDB is considered a prediction, is considered in the training. I didn't mention exactly how that training works, but basically the training involves generation of parameters that lead to structures that look like the proteins in the PDB. So this means that if there are some systematically poor structures in the PDB, maybe in some category of protein, then you're going to get back those systematically incorrect aspects. So you don't expect it to be perfect. Also, there's not that much information about residues that are far apart. And this is part of the simplification that goes into AlphaFold. It basically ignores residues that are very far apart in sequence so that to make the process practical. And that results in models that may have distortions, and you'll see that, and also incorrect domain relationships are common. So keep those things in mind when you are going to use your AlphaFold models. So here's an example of a good AlphaFold prediction that we're going to look at in just a little bit of detail. So this is work that Tristan Kroll did on PDB entry 6TE3 that he was working on. So here's some density from an X-ray map, and look at that leucine 710, and you can see there's a dark blue model, and that's the AlphaFold model. And ask yourself, does that AlphaFold model fit the density? Well, superficially, sure, kind of goes in the right place. But if you look in detail there, not really. The blue atoms are out of density. And if you look at the other blue atoms around that structure, they're often a little bit out of density too. And this structure is pretty high resolution. So it actually is telling us something about where those residues really go. This is a high confidence AlphaFold prediction. I didn't give you the scale, but here the scale is 0 to 1 or 0 to 100. And when you get close to 1 or 100, depending on your scale, that means pretty much all the C alpha atoms are in pretty much the right place, very close, within half an angstrom or so. And indeed, they're in that kind of ballpark here. But still, the side chains are not all right. So Tristan rebuilt that model, starting with the AlphaFold model, and with very little changes, in this case at 710, putting in a more common rotamer, suddenly it fits the density a lot better. And similarly for the other parts of this structure. So the details may not be quite right, and keep thinking of AlphaFold models are great hypotheses. So here's an example of the long distance relationships. Here's a structure, 7BGL, that was part of the CASP14 structure prediction competition. And this is just one chain shown as it appears in the PDB, domain 3 of chain A, makes a big ring. So here is the AlphaFold prediction. Wow, doesn't look quite like it. if we look at contacts, we'll see that, well, doesn't look all that great. You know, one protomer that's like overlapping completely with another one. So this is not uncommon in AlphaFold predictions of large molecules, that longer distance relationships are not so good. But if we just look at one monomer, wow, that's not so bad. It's not perfect, but it's in the right ballpark. So this was a pretty good AlphaFold prediction locally, but globally it was pretty terrible. Once again, AlphaFold models are great hypotheses. So let's summarize this. What can we expect from your AlphaFold model? And I mentioned AlphaFold here, but Rose TTA Fold, other programs and software that's going to appear in the future are all going to have similar kind of characteristics if they have similar inputs. And as more sophisticated inputs are used, they're going to overcome some of these limitations. So they're great hypotheses. Save that in your mind. Parts of them can be amazingly accurate. Parts of them are totally wrong. And the confidence measure is helpful, but it doesn't fully reflect accuracy. You can numerically quantify that, as the AlphaFold people did. The correlation, Pearson correlation coefficient between the confidence measure and the actual value of that LDDT is 0.75. And 0.75 means things are generally in the right direction, but they aren't always exactly right. So the confidence measure is helpful, but not perfect. Okay, so what can we do with these? A lot. We're going to jumpstart structure determination, and I'm going to give you a couple examples of how to do that. And everything that I'm talking about today, you can do today. So what about AlphaFold models makes them good for jumpstarting structure determination? Well, one thing is this confidence measure I told you about a moment ago. So if the confidence measure is pretty good, which it is, it allows you to know kind of in advance what part of models to get rid of, because if it's low, you know you don't know the answer. So you can trim or prune your models to get rid of the worst parts. Then the high confidence parts, as we've seen, are often very accurate. We can often just use those as a great hypothesis. Something that's a little subtle is that an AlphaFold model is better than a homology model because you don't have to worry about insertions and deletions in the sequence. The sequence is automatically correctly assigned to the right part of the structure. And if you had a homology model, sometimes you might have a mistake in sequence alignment. There aren't any mistakes in sequence alignment to speak of in these models. So that's really great. That means if you place the beginning and you place the end, those are good. And then you know exactly what residues go in between. Okay, so here's an example. This is a real life example. Jamie Wallen at Western Carolina University sent us a help a year ago with this structure where it didn't have great geometry, and he wanted help from Pavel Afni. And Pavel gave it to me, and neither of us were able to really solve this problem. We got it a little bit better, but didn't solve it. Okay, and then fast forward a year later, we get to AlphaFold, and I asked Jamie, can we try this again? And so, yes, he said, let's do it. And so before AlphaFold, the model here is, okay, this is a 2.6 angstrom SAD structure refined against 3.1 angstrom X-ray data. RR free 2729. It's not terrible, but it's not really good for this resolution. And there's a big problem that there's some residues that are missing in the model that you can see with the dotted lines. And here's the density. And okay, have a look at that density. Can you figure out how you connect residues 118 to 128 and how to connect residues 139 to 147? Think about it, and we're going to see what AlphaFold suggests for you next. So here's that model. Let's just go and generate a new AlphaFold model based on the sequence. So we just do that. Go to AlphaFold. We have a server that you can use. You can use any of the servers and get pretty much the same answer. And here is the AlphaFold model. Wow, look at that. It superimposes on a lot of the structure that Jamie created. And it presents a hypothesis for us about how to connect 118 to 128 and 139 to 147. You see that this hypothesis says that we got the end residues a little bit wrong, and we connect through the density. Wow. So the AlphaFold model didn't use that density, but it puts those residues in pretty much as good a place as you or I or anybody could figure out for fitting that density. So Jamie liked this and refined the model from there. And now after AlphaFold, the RR free is 2124, which is in the right ballpark for your 3.1 angstrom structure. And what does this say? It says, yes, this was a good hypothesis. Didn't have to turn out that way, remember, but this one was a good one. So it's a great hypothesis. All right, so what about molecular replacement? That's the other big technique in crystallography. Well, Tom Flower and Jim Hurley wrote a paper earlier last year on this. And here's a summary of their work. So they had submitted a structure to CASP as well, which ended up being in 7JTL, has 2 chains, and the yellow and gray chains are superimposed in this little picture. And the AlphaFold model that they generated is in blue. And you can see that a lot of this model superimposes, but parts of it do not. They showed that you could take that model with molecular replacement in 2 angstrom data that they had, and parts of it are amazingly good, and parts of it are not quite so right, with side chains in wrong positions in a number of cases. And sometimes even the main chain needed rebuilding. So the message there again is AlphaFold models are great hypotheses. Okay, what about Cryo-EM? And we're going to come back here to the example structure that we've been working on, which is 7MJS. And we already saw this picture. This is the showing here now, the upper right part that was an AlphaFold model prediction that wasn't that good, and the lower left that we're not going to look at the moment, that was quite good. So what can we do with this? Well, we have these great estimates of error, of uncertainty. Let's not call them great, very good. Estimates of uncertainty. And it says that this part is all uncertain. We saw that in that picture early on. Well, let's just get rid of the bad part, of the low confidence part. So here's our model now for most of the structure. Let's take this model and just dock it straight into the density map. Wow, it just fits right in there. It's amazing and is really good. So now we've docked it. Let's trace the loop based on the density that's present there. This is a pretty nice map, not so hard. And what do we get from that? Here's our AlphaFold trimmed model. And here we've generated a rebuilt model based on tracing through that density. Now it looks a lot more like 7MJS. Notice that this rebuilt model doesn't look perfect. And we'll come back to that in a moment and see how to make it better. So that brings us to our third part. We can make AlphaFold prediction better if we have some experimental data. So here's that picture we just saw. We rebuilt this model using our density map, and now we have a new model. What if we use that rebuilt model as a template in AlphaFold prediction? So AlphaFold is able to take information like the backbone positions or the backbone positions of another structure. And actually, as a default in many systems, AlphaFold prediction goes to the PDB and finds all the similar structures, all the similar sequences, and uses those structures as templates. That's a common method. Well, suppose we use as a template our rebuilt model. And the question is, can we get a better AlphaFold model by doing this? So let's do that. And let's think about how this is working. Our initial AlphaFold prediction started with a sequence, multiple sequence alignment, and all those parameters gave us a prediction. So now we're going to add in, the prediction is not that good in the loop region. And now we're going to add in a template. And we're not going to use the template, AlphaFold does not use the template directly as this is the structure. Rather, it takes that template and says, what are the backbone phi-psi angles? And it uses those as information about a possible structure. And it also indirectly uses the information about what is close to what in this template to allow it to focus attention. So it's going to use those backbone angles, and it's going to focus attention on residues that are close. And in the prediction, the initial prediction, it wasn't clear what residues are close in that loop. But now we have a better idea about that. And you get a new 3D prediction. And wow, that prediction is very nice. The template definitely improves the prediction, but more than that, the new prediction is better than the template. You can just see it there. The beta strands make a nice little two-stranded loop, and it's beautiful, whereas the template wasn't all that good. So we improved the prediction, and the prediction is better than our template. That means we can iterate this. Let's look at this now in just a little bit more detail. So here's the same structure, the same map in a little bit of detail. And this is the initial AlphaFold prediction, just at the place where it starts getting bad. So the left-hand side is good. You can see the model, once superimposed, fits the density map quite well. And keep in mind, these AlphaFold models, we're just taking the AlphaFold model, which is in any orientation, in any position in space, and we're just going to superimpose it on our density map. And we're not going to adjust that AlphaFold model in any other way. We're not going to refine it or anything. And it just, boom, fits right in there on the left-hand side. The right side does not look that good, right? It's clearly not doing the right thing. So now we're going to rebuild that model. This is the rebuilt version. And this rebuilt model is actually not that good. You see that little arrow there. It's got some really bad geometry at the junction, but it kind of is starting to fit in the density. So I see a question now. I'll answer that one now. Is prediction of membrane proteins good, and is it taken into account? So the membrane is only implicitly taken into account for membrane proteins, implicitly because the training set involves the membrane, but it's not explicitly done. And prediction of membrane proteins can work, but it can also be not that good. Okay, so back to this example. So our blue model is not that great, but it's starting to get in the density. And now we're going to use the blue model as a template to create a new AlphaFold model. And here's that new AlphaFold model. It's also not quite right. It's fixed up that little bad place where the arrow is. It's not all in density, but you'll see that if we rebuild this, the rebuilding doesn't have to do very much to pop it into density. So the AlphaFold model was very close. It's just a little teeny rotation there. And then we're into density. Now we use this as a template. And there's our new AlphaFold model. Doing another cycle. There's our final AlphaFold model. And this AlphaFold model matches the deposited structure very closely. Now, don't get the idea that this is always going to work so perfectly, right? This is a great example. It has high resolution data, but this is what is possible. And this is what happens with a good data set. And once again, note that that AlphaFold model is superimposed. It's not refined. This is just plopped right down into the density. Boom, it really fits. It's amazing. So what have we learned from this? You can rebuild an AlphaFold model if you have a density map. The rebuilt model improves the next AlphaFold prediction, and you can improve that, iterate that to improve the model. You can read about that in the preprint that is listed at the bottom of the page there. This preprint comes along with data as well. So it involves running 25 different structures that were deposited in the PDB after AlphaFold was created, after the AlphaFold database was created. And all the data from all the structures are there, including instructions on how to reproduce that data and Chimera X sessions so that you can see all the results. And so you can evaluate exactly what's happened for all these cases yourself. And none of these were selected. I just took, at the time of running this, I took the 25 most recent structures that had between 200 and 1,000 residues that had CryoEM data that were suitable. So basically, this is what you get on average for these. One thing to keep in mind about that test is that we tried to simulate a situation where you don't have similar structures in the PDB. So we did not include templates from the PDB in those predictions. And similarly, in this example that I just showed you, we did not use templates from the PDB, of which there could have been. Actually, I didn't look to see if there are, but there probably are templates for this one in the PDB. So we didn't use those to simulate a situation when you don't have them. Okay, so now, given all this, how should we solve our structures today? So let's run through this, and then it'll be time to try to answer any of the questions that haven't been answered yet. So let's think first about how are we going to get the best AlphaFold or other prediction method model given what information we have. So we have our sequence, and we have our multiple sequence alignment, which we can make ourselves, or we can let the program do it for us. We have templates from the PDB, potentially, and we have templates that we have created. And maybe at the beginning, we don't have any of those, but maybe we do. Maybe we already have a model that we've built, and we're just trying to make it better. So that's a way to think about all these things. So those are the kind of things you're going to have as inputs. And with those inputs, AlphaFold or other prediction programs will generate a prediction. Great. And then once you've got a prediction, if you have experimental information that allows you to rebuild it, like a density map, you might rebuild that model, and maybe you would fix errors as much as possible. Maybe that rebuilding was done automatically, and then maybe you went in and fixed some errors, and then you got a rebuilt model, and then you took that rebuilt model, and then you put that in your list of templates that you have created, and you can run the whole thing over again. And then if you have improved the model on the right side, then you can expect that the new prediction should be better. And so what tools do I have to do this today? Well, AlphaFold, you can run that on Google Colab, and there are many tools to do that. In our Phenix software, you can press a button on the GUI and it will pop up a window that will allow you to do this, for example, but that's not the only place. You can do it anywhere. Once you have got a prediction in Phenix, you can use dock and rebuild to dock that model automatically and rebuild it. Other software will do that for you too. You can fix your errors with Isolde, with Coot, with any way that you want, and then you can iterate that process. So now what about doing this in Cryo-EM? Well, it's pretty much what we just saw. In Cryo-EM, we have a density map, and we're going to get an AlphaFold prediction for each chain in our structure. Now, if you already have a model, like if the exact structure is already in the PDB, you don't have to use the AlphaFold prediction, right? You can use something from the PDB if you want, but you don't have to either way. Now, so if we have our density map and an AlphaFold prediction for each individual chain, in Cryo-EM, the map is more or less independent of each model. So you can independently rebuild each AlphaFold prediction one at a time. That leads to a model for each chain with a full exact sequence that's been rebuilt by you. Then maybe you're going to fix the errors, refine the model. So now you're going to put everything together, and you have a refined model for each chain. You're going to then run AlphaFold with the refined model for each chain to get a new prediction. So the first step, once again, we can do that with dock and rebuild or other automatic procedures for rebuilding an individual chain. You can fix your errors with refinement or interactive tools like Isolde and Coot. And Colab will give you a new prediction. So you can cycle through this once, 2, 3, however many times you wish. As long as it's getting better, you might want to do that. Now, crystallography. So this is the last slide, or last work slide here. The workflow for crystallography is a little bit more complicated than for Cryo-EM, because in crystallography, improving the map involves requiring all the components of the structure to be present in place at one time. So in Cryo-EM, you have a pretty static map, and so you can work on one chain at a time, pretty much. In crystallography, you're going to want to work with everything at once. So that means we have X-ray data, and we have a prediction for each chain or a homology model from the PDB, whatever you like. And then we're going to do a molecular replacement with all the components. And we're going to get a molecular replacement model that has all components. Then one component at a time or all together, you might do automated rebuilding. So to do them all together, for example, in Phenix, you would use Phenix auto build with that entire molecular replacement model. Once you have an automatically built model and your density map, then you can bring in your AlphaFold prediction and improve each model. So now we're going to take the AlphaFold prediction for chain A and our auto-built model for chain A and our density map, and then we are going to put those things together to try to make the best possible model. And we might do that by hand, or we might do it automatically. So we're going to rebuild the AlphaFold predictions based on our auto-build model. And the reason for doing it in this slightly convoluted way is the result is then a rebuilt model for each chain that has the full exact sequence. So now we've created a new model for each chain that has all the right residues and hopefully a lot of them are in the right place. And now we can refine all of those together, and then we can go back and get a new AlphaFold prediction. So a few little steps in there for the crystallography that are slightly more complicated than Cryo-EM, but still not that tricky. And you already have tools to do everything. You can do molecular replacement with phaser or other tools. You can do the automated rebuilding with AutoBuild or other automated tools. You can rebuild with dock and rebuild. You can fix errors with Coot, Isolde, Real Space Refine, and you've got AlphaFold Colab to go around the circle. Okay, so what have we seen? So number one, and most important, AlphaFold models are great hypotheses. Number two, they're great for starting structure determination. You can improve them. And last, they allow a new workflow that at the moment is slightly more complicated in that it is iterative over everything that you do already. I'm sure we and others are going to make this more automated to make it easier down the road. And in the meantime, you can already do everything that I just showed you. And thanks to you for your attention. And thanks to all the people that are involved in the Phenix project and around the world who are working on similar things as well. And I'd be very happy to take these questions now. Maybe Jean can help me sift through what we've done. Thanks.
Okay, so you have many questions in the discussion panel. You can see them.
Okay, let's see.
Let's see.
So let me start with a question that's actually in the Q&A over here from Marco Petrone. By comparing AlphaFold and RosettaFold, I find that AlphaFold tends to predict more secondary structures and to not show cross-domain interference in chimeric artificial proteins and sequences. So the two moieties seem predicted separately. Is that good, or they're just two different types of bias? Yeah, that's a really good question. And maybe a good way to think about this is back to our AlphaFold models and RosettaFold models. They are hypotheses. And so it's good to generate these models in as diverse a way as you can, so as to have a diversity of hypotheses to consider. So I wouldn't think of these as RosettaFold's better, AlphaFold's better. Think of it as, here are a number of hypotheses that may or may not be compatible with the other information that I have. Let me look at all these hypotheses, evaluate which ones are consistent with my data, and then use that to limit the number of possibilities of what I think is actually happening. And it may be that your data cannot distinguish amongst some of these hypotheses. And take that as a positive. If your data don't distinguish between two hypotheses, that means that those hypotheses, either one may be correct. And so think of those as information about what you do not know. So that tells you something about the range of possibilities that are compatible with your data. Okay. So Jean, maybe you can point to some of these questions that have not been answered by somebody else.
Let's go back to the list, discussion list. So there was a question from Gerard about the beta strand. That was on one of your slide.
Okay.
This is a question. This is the second one. The beta strand arrow is pointing from 128 to 118.
Oh,
Yeah.
that is interesting. I could have labeled it wrong, or it could be that it's the other way, but it goes the right way. Actually, there's no question about it. So I must have labeled it. I'll have a look at that, Gerard. Thank you for pointing it out.
The incorrect domain relationship still a problem with AlphaFold Multimeter?
So I don't personally have a lot of experience with AlphaFold Multimeter, but I would expect it would be pretty much the exact same situation. That so within one chain, the relationship between domains is often slightly or sometimes greatly incorrect. So I would expect that that problem would remain with AlphaFold Multimeter. In some cases, it might be reduced if that long distance information about the multimers is used. But as you saw from my prediction of the 7BGL, multimers are not necessarily done all that well.
Can we predict peptide binding site?
So there are some recent papers on that. A lot of excitement about binding of peptides. I haven't done it, but others have done that. And so there's a lot of work on that right now. Yes.
And then one more. I mean, can we use AlphaFold to predict mutant of a native protein? I think this one is.
Yeah, that's a great question and a very important one. So the basic answer is no. A number of people have tried to do this, put in mutant sequence and ask, do I see a change in my structure? And basically what you get back is you get the new residue is different because it has to be, and the rest is pretty much the same. And the logic that people are using to explain that is that, well, AlphaFold is going to use your mutant and then all the other structures to predict the structure of this one. And the structure of the mutant is going to be really similar usually. And so it's going to give you pretty much the same answer. To expand on it is just a little bit, AlphaFold tends to give you one answer, even if there are multiple actual structures that are consistent with a particular sequence. So if your structure actually takes on multiple conformations, AlphaFold will typically give you one of those. Even if you run it multiple times with different parameters, it gives you pretty much that one back. So that's a limitation of AlphaFold. And some people have tried to get around that with some success by manipulating the multiple sequence alignment. So the multiple sequence alignment tells AlphaFold in principle about what's close to what. That's what we learned. Now, what if half of your structures, half of the time, it's in one conformation and half the time it's in another conformation. And the residues that are close, one in this set of structures is different than the residues that are close in that set of structures. All right, so if you do a multiple sequence alignment, you're going to see correlation between covariation between residues that are even just in one half of that set. So AlphaFold could get fooled that way. Now, if you were very clever and were able to separate your multiple sequence alignments into the group that's conformation A and the group that's conformation B and submit that to AlphaFold separately, you might get a very different answer. So that kind of logic is how people are trying to work around that problem.
Okay, so is this approach AlphaFold we build, AphaFold we build better than traditional refinement for X-ray structure? Molecular replacement, we build, we find, we build, we find.
So the answer is yes and no. So let's suppose you have a highly refined X-ray structure. You have a 2 angstrom structure. You've worked on it a long time. You got everything fitting just perfectly, and you have a great R factor, and geometry is really, really nice. Okay, you do everything I just told you, and AlphaFold will come up with a model that's basically the same as your model. And so you're not going to make it better if it's already pretty much as good as it's going to get. So AlphaFold's not going to beat a really great, already done X-ray structure or Cryo-EM at high resolution. That being said, if you don't have a great structure, then refinement, including AlphaFold in the way we're talking about here, that you take your model and you feed that to AlphaFold and you get a new prediction and you refine that AlphaFold model and fix all the mistakes in it, that can improve your structure. Keep in mind that AlphaFold may continue to put side chains in wrong conformations, even if you give it the right ones, because it doesn't notice the side chain conformations that you give it. It actually only is going to use up to C beta. So it doesn't even know what the side chain conformation was. It throws it away. So even if you keep fixing a certain lysine, it's going to put it back where it wants to go, or not necessarily where you put it. So you cannot just take that AlphaFold model ever and just use it as the answer. The iterative process that I described is going to give you a better and better AlphaFold model that requires less and less of your work at the end to fix up. But don't think it's done. You or your automated tools or something still have to work on that model to make sure that it is compatible with your data.
I mean, what about using PDB redo structure, which are better than the PDB model for training AlphaFold?
I think it's a great idea. And retraining AlphaFold with curated data sets, with PDB redo, with other better structures, iteratively redoing it with AlphaFold, all these things are great ideas, and they will be done. These are things that are going to happen. You do have to keep in mind that training these programs is not an afternoon affair. It's something that involves a lot of work. There's a group that's making an open AlphaFold, and they have made a lot of progress on that, and they now have a working version that is trainable. They have not yet reported doing it, but I expect that to happen pretty soon. And once that's available, then others can use the software to retrain AlphaFold, generate new sets of parameters that are suitable for this situation or that situation, that are better than the ones that exist. And that can iteratively go on, and it will. And I think that's all fantastic.
What about nucleic acid structure? You know,
that one's going to be, that's not there yet, right? So AlphaFold does not know about nucleic acid structure. And there are tools that people are working on for machine learning prediction of nucleic acid structure, not quite at the same level yet. There's a lot of information in nucleic acid sequence as well. But it's not obvious to me that it's going to be as clean a problem as it is for proteins, partly because there are not nearly as many structures to learn from for nucleic acid structures compared to protein.
Okay, do you think there is a limit on the size of the sequence which can be used for prediction?
So currently the limits on size have to do with memory requirements for running these programs. So AlphaFold, if you run it online with the collab, basically you can get things up to about 1,200 residues, something like that.
Mm-hmm.
Beyond that, you start running out of memory that's available to you. If you have your own GPU, you can do bigger structures, but it's still not unlimited.
A small question, can you recommend some program for fitting in CryoEM map? What is the question? I've lost it. Which program do you advise to use to rebuild, optimize a model, or it's docking into an EM map?
Oh, there are numerous programs you can use for that. So in our Phenix software, you can use Dock and Map to do that. You can do that kind of thing in Chimera very readily. Isolde will do that. CryoEM programs can do that. Code can do that. So there are many tools that can do all the things that I've described.
I mean, there's a question about the parameters. You give a brief can you give a brief classification or type of those millions of parameters that are used by AlphaFold?
Not quite sure exactly how to answer that question. So AlphaFold creates a database. They created a database of these 21 million parameters. Those are parameters for all the inference engines that are in AlphaFold. So basically, all the things that could be adjusted to give a better prediction are in those 21 million parameters. If you ran a new prediction and you changed one structure in the PDB, basically all 21 million parameters are going to change.
Okay. Let's see, go down. Is Alphabet making business with AlphaFold? So I'm not sure to understand the question.
Well, I can just briefly address that. So the AlphaFold people have started a new company, and the purpose of that company is to use AlphaFold and similar new generation software to solve major biological problems, including drug discovery. So yes, they're trying to go ahead with that. But from the academic point of view, the good thing is using AlphaFold is free and totally open. The source code is completely open. You can get it, you can change it, you can do whatever you want with it. Their parameters are non-commercial only. So you can't use AlphaFold to predict something today if you're a company, unless you talk with Google.
And what about IDP, non-structured protein?
AlphaFold is great for predicting IDPs. So basically the parts of models that AlphaFold predicts that it doesn't know anything about are very often IDPs, parts of proteins that are not structured. So that's actually a really great thing about AlphaFold, in a negative way, that if it doesn't know, that's probably what's happening. Not always. So basically a better way to say it would be, if you have a lot of sequence homologs, so a lot of multiple sequences in your multiple sequence alignment, and it says it doesn't know anything, good chance, not sure, but good chance that it's unstructured.
Do you have plans to implement multimeter modeling within the Phenix Collab notebook?
In principle. So I haven't done anything with that yet. We're certainly thinking about doing that. I haven't figured out exactly how to proceed with that versus working on making the notebook work more automatically for this rebuilding and iteration of the whole cycle. So we may do that first and then come back to multimers later.
Okay. Good. So maybe what we can do, if people have more questions, they can send you the question directly.
They sure can.
You will
They can send, anybody can send me emails. I'm happy to answer them, and happy to answer questions about using software to do the things that I've just talked about.
So thanks, Tom. It was a great presentation, sir, and thanks everybody for being here today. Have a nice day.
Okay, thanks
Bye.
everybody.
Bye.
Bye-bye.
Okay, it was good. Quite good. Thanks, Tom. Too many questions.
It was great. Thanks a lot.
It was great.
Thanks for inviting me.
Yeah, it was great. So many questions.
Yeah, a lot. You had a lot of questions there. Yeah,
And still
that's great.
of them. Okay, thank you.
Okay, see you later.
See you soon.
Okay, bye-bye.